Bismillahirrahmanirrahim. Now I am going to discuss about structure of DNA. Uh, this is the nitrogenous base. This is ribose sugar, 5 carbon containing ribose sugar. And so nitrogenous base with ribose sugar is called nucleoside. If we add phosphate with this fifth carbon, it is called nucleotide. So nitrogenous base ribose sugar is nucleoside. If when we add phosphate at this carbon is called nucleotide. So you follow that here phosphate is added or phosphate is present. So this structure is nucleotide. Above the structure is nucleoside although, but here there is phosphate, but I didn't show here on the phosphate, but here phosphate present. That's why it is called nucleotide. Okay, I am drawing another phosphate. Just a minute. Okay, I show you here phosphate. So it is nucleotide. Nitrogenous base, ribose sugar, and phosphate. It's called nucleotide. Now, if we make a bond in between two nucleotide, here one nucleotide and another nucleotide. If this one is another nucleotide. If we want to make a bond in between two nucleotide, we will get a bond name is ester bond, and in between two nucleotide we get two ester bond, that is why it is called diester bond, and this bond is formed by phosphate, that's why it is called phosphodiester bond. So ester bond uh, with this phosphate is called phosphodiester. It means diester means two ester bond. Here is one ester is formed and another ester bond is formed, and in between the ester bond there is phosphate, so phosphodiester. How this diester form is formed, now I am going to discuss. Here you follow. We already know ester bond is formed between two substances with removal of water. One molecule of water is removed in between two substances. So ester bond is formed. Ester bond means reaction between two substances and removal of one molecule of water. So nucleotide, this nucleotide reaction occur and water is removed. Here you follow 1, 2, 3, third number carbon. Here HO or OH is removed as water with this hydrogen. So one hydrogen, two hydrogen, and this oxygen is removed. Here you follow OH with this hydrogen is removed as water. As we know, phosphate has this H3PO. Suppose H3PO4. So PO4 means four oxygen and H3PO4. 1, 2, 3, 3 hydrogen, H3PO4. PO4 means 4 oxygen. 1, 2, 3, 4. Here oxygen, we know oxygen has double bond. So double bond. So 3 hydrogen and 4 oxygen. H3PO4. Now follow, in between nucleotide there is phosphate or H3PO4. So this is the chemical structure of H3PO4. Now this hydrogen from this H3PO4 with this OH of the ribose sugar is reacted or reaction occur and water is removed. So here ester bond formed. Here same reaction occur. Hydrogen of H3PO4 and OH of ribose sugar is reaction occur and water is removed. Here also ester bond form. So double ester, and due to presence of phosphate which is called diester, phosphodiester. So after the chemical reaction we get this chemical structure. You follow, two hydrogen is removed, so this structure is formed. Oxygen, there is oxygen, hydrogen is removed as water, and this OH is present. Due to as this OH was present, so we can get this chemical structure. I think you are clear about phosphodiester, phosphate and double ester. Here you follow that one ester bond is formed and another ester bond is formed due to removal of water and presence of this phosphate. So phosphodiester. This is the very much easy. Now in the next I will discuss about 5 prime to 3 prime. Here, you follow that direction of the first direction of the carbon number direction. You follow that five. This is the fifth number carbon. 5 4 3. Third phosphate. This phosphate and this ester bond formed at the third carbon. This ester bond formed at the fifth carbon. So direction of the reaction is 3 to 5 prime. Direction of the reaction is 3 to 5 prime, but chain elongation or polymerization occurring 5 to 3 prime. Here follow 5 carbon, third carbon, but reaction between 3 and 5th carbon. Phosphodiester formed between 3 and 5th carbon or third and 5th carbon, but polymerization occurring 5 to 3 carbon direction. 5 3 5 3. So polymerization occurring at 5th to third carbon, but reaction occurring third to 5th carbon direction. I think you are clear about this. You follow here that I draw this DNA double helix structure. This is nitrogenous base, this is phosphate end, and this is the ribose sugar. I just draw as a structure of DNA. This is the on helix or on DNA backbone on side, and this one is another helix. We know DNA is double helix, so you follow polymerization. This is the nucleotide. This is the phosphodiester bond. Nucleotide are connected by phosphodiester bond. I already discussed how phosphodiester bond is formed. So phosphodiester bond or this polymerization occurring 5 to 3 prime direction, and another side helix is opposite direction, phosphate. Here phosphate is above out at the top, but at the down phosphate. Here sugar end is at the down. Here sugar at the upside. So just opposite position of the nucleotide. Phosphate is downside, upside at the sugar, and nitrogenous base is same side. We know adenine and thymine are bonded by two. In between adenine and thymine, 2 bond present, and cytosine guanine is 3 bond present. Hydrogen bond, these are just hydrogen bond, 2 hydrogen bond, 3 hydrogen bond. Cytosine guanine or guanine cytosine is 3 hydrogen bond. Adenine thymine adenine is 2 hydrogen bond. 3 hydrogen bond between cytosine guanine or GC, CG, GC. ATA is double hydrogen bond. This hydrogen bond is weak bond. If we want to separate this, it will be it is very easy to separate this hydrogen bond. So direction is one helix direction is 5 to 3 prime and another helix direction is 5 to 3 prime, but 5 to 3 prime is from above to down and from down to above. Just position of the phosphate ribose alteration occur. Here phosphate at the top and downside is ribose sugar. Here phosphate at the down and ribose sugar at the top side. This is the chemical structure of DNA. Thank you.